This video describes a procedure to generate Carcinoma-Associated Fibroblasts, or CAFs, from primary cultured human mammary fibroblasts. Carcinomas are complex tissues comprised of neoplastic cells and a non-cancerous compartment referred to as the stroma. The tumour-associated stroma consists of extracellular matrix and a variety of stromal cells, including fibroblasts, myofibroblasts, endothelial cells, pericytes, and leukocytes. Carcinoma-associated fibroblasts, rich in myofibroblasts, a hallmark of activated fibroblasts present within the tumour stroma, play a major role in driving tumour progression. These can be generated in vitro by first isolating primary human mammary fibroblasts from healthy breast tissue obtained by reduction mammoplasty. The obtained tissue is dissociated into a single-cell suspension. Then immortalized by viral transduction and engineered to express GFP and a puromycin resistance gene. The cells are then co-injected with human breast carcinoma MCF7 RAS cells subcutaneously into an immunodeficient mouse. The injected human mammary fibroblasts will evolve into tumour-promoting CAF myofibroblasts. Then the parental human mammary fibroblasts are extracted from tumour xenografts and cultured in the presence of puromycin. The resulting puromycin-resistant cells are called experimentally generated CAFs, or EXP CAFs. Immunohistochemical analysis reveals that initially